Hello, we're glad you joined us for this live webinar, Integrating Genomics Platforms and Data in Biobanks. I am Christina Mahalik of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. To learn more, visit labroots.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left of the screen. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left corner of your webpage and follow the process of obtaining your credits. I am now delighted to present today's speaker, Sami Amr, PhD, Director of the Translational Genomics Core Partners HealthCare Personalized Medicine, Harvard Medical School. Sami Amr is the Director of the Translational Genomics Core of Partners HealthCare Personalized Medicine, where he feels privileged to work with basic and translational researchers across Partners HealthCare to identify DNA, RNA, and methylation markers and signatures of diseases that can help deconvolute underlying mechanisms of pathogenesis, as well as being leveraged in diagnostic and clinical assays. Dr. Amr will now begin his presentation. The floor is all yours.
Thank you very much, Christina, for that introduction. And thank you for inviting me to present this presentation, this webinar. And essentially, what I'd like to talk to the audience today is relating to how we can integrate genomic data into biobank platforms. This slide shows my disclosures. I'm an employee at Partners HealthCare Personalized Medicine, which offers research and clinical genomics services in an academic and not-for-profit setting. And for the majority of this presentation, I will be sharing with the group and discussing our experience in integrating genomics platforms into the Partners HealthCare database. And there's two major objectives for this presentation. The first objective is to be able to describe the different genomics platforms and technologies that could be leveraged in DNA plasma or serum biobanks. And the second objective is to also explore strategies that allow easier integration of that genomics data into the existing infrastructure of the biobank in a way that promotes accessibility to data for researchers, as well as allows for ease of use of data querying. So to begin this discussion, I'd like to just give you an overview of the Partners HealthCare Biobank. The Partners HealthCare Biobank is a repository of consented patient samples that is linked to the electronic medical records of these patients and is supplemented with health information as well as family history from surveys that are provided to the patients. So these are essentially self-reported surveys that are collected. And today we have more than 57,000 patients in the Partners HealthCare system have consented to participate in the biobank. And we've actually done blood collections for more than 41,000 patients. And currently our target is to consent 75,000 patients by 2018. The two major objectives of the Partners Biobank is to advance, is to accelerate research by allowing immediate access to samples and data to researchers. And the second one is to reduce the cost of research, and so that the per sample or per patient inclusion into a research study is significantly less expensive than it would be if a researcher was to do it outside of the biobank. So both of these objectives are meant to drive medical research in order to improve healthcare. And essentially, one can look at this as the biobank allows for accessibility to large patient cohorts, to medical information, as well as easy access to different sample types from these patients. What our biobank provides are consented DNA, plasma, and serum samples. And if a researcher is interested in one of these sample types, the cost per sample would be $20 to $35 per sample. However, the estimated cost if a researcher was to collect these sample types outside of the context of the biobank, it is roughly between $300 to $1,000 per subject. So clearly, there are major incentives to researchers working within the Partners HealthCare system in being able to access biobank samples as well as access to biobank patient information. This is just a general outline of the operations of the Partners HealthCare Biobank. Essentially, there's 4 different components or phases. The first phase is recruitment, and the recruitment of patients is either done in person at the hospital during their clinical visits, or patients also have an option to consent electronically through the Biobank portal system. The second phase, or the second piece of the second step in the biobank operations workflow is phlebotomy or blood draws, and both research and clinical phlebotomists can draw blood from patient samples. We also collect samples from clinical discards. The third component is sample and data management. And this is essentially a large IT and bioinformatics infrastructure to be able to track samples coming in, link them to medical health records of patients, as well as any genomics type data that is produced in downstream applications. And the final piece of the biobank operations is, of course, sample and data distribution. Essentially, workflows that we've implemented to be able to deliver samples back to different researchers that they're interested in, as well as be able to deliver different types of data, whether it be medical records data or genomics type data, back to investigators that are interested in the biobank, the different biobank cohorts. So what exactly does the biobank provide investigators with? It provides essentially access to patients and cohorts that are consented for broad-based research, as well as patients that are consented for return of actionable results. In addition, these patients are also consented for recontact for additional info or additional sampling or inclusion into different studies. Investigators also can access different types of sample types. Essentially, the main sample types that we extract out of the blood draws are serum, plasma, and DNA. We work with collaborating studies, investigators that have their own kind of cohort analysis, and we can collect 2 additional types from these collaborating studies that are incorporated as part of the biobank sample repository. And the other piece is that investigators have access to discarded tissue samples. And all of these, as I mentioned before, are immediately available and are available at a much lower cost than they would if if they were collected outside of the biobank system. The types of genomics data or medical records data that is provided to investigators. Essentially, right now we are offering genomic data for 25,000 patients. In addition, this information is linked to the electronic medical records of these patients, as well as the self-reported surveys and data on family history, lifestyle, and environment. And finally, we've also supplemented all of these different, services that are part of the biobank with powerful and state-of-the-art query tools that allow you to essentially use the EMR data, electronic medical records data, as well as the genomic data to be able to identify different cohorts, to accurately identify a cohort with a disease of interest, as well as identify controls within the population for a given disease. So what types of health information are linked to these samples that are found in the biobank? There's 2 different major types of data. One is the clinical data, and this includes both coded data as well as text data. Coded data includes essentially diagnoses based on ICD-9 or ICD-10 codes, demographic data that is provided within the electronic medical records, as well as any laboratory test data, as well as medications data for a given patient. Text data is essentially the notes that are physicians that are included as part of the electronic medical records that are provided by the physicians, discharge summaries, operative reports, cardiology reports, or pathology reports. And the second large component type of data that is provided as part of the health information that is linked to samples is survey data. And those are self-reported surveys that look at or query family history, environmental circumstances, as well as any other kind of lifestyle related type data. So what are the characteristics of the participants of the Partners HealthCare Biobank? Essentially, it pretty much reflects the population that is found in the Northeast and in Boston. The majority of individuals that are included in the biobank are Caucasians. 83% are white. We have a 58% to 42% female to male ratio. The majority of patients are closer to the 50s and 60s age categories or age groups, and about 58% of the biobank participants have either a college or graduate degree, so it's pretty reflective of the population that is unique to Boston and unique to the Northeast. And based on the report on the ICD-9 codes that are found in the medical records, this table represents the different types of phenotypes that are identified in patients represented in the biobank. And the highest frequency phenotype that is reported is joint pain. However, there's a broad range of different types of complex diseases that are found in these patients, including, for example, hypercholesterolemia in about 13,000 patients, ischemic heart disease in about 7,000 patients, as well as psychiatric disorders, including depression in about 6,600 patients, and a range of different kind of complex diseases that have some genetic link or have some kind of polygenic effect. In addition to trying to identify the patient phenotypes based on the ICD-9 codes, we've developed and incorporated an algorithm that is able to accurately predict the diseases of different patients based on the information that's found in their electronic medical records. And these are validated phenotypes with a high predictive positive value for that phenotype based on the algorithm that we've developed for these different types of diseases. So this information is also available to researchers, and researchers can access this information to identify a more accurate cohort rather than rely on the ICD-9 or ICD-10 codes, which are not always reflective of true diagnoses. In addition to that, we also use the Charlson Index to be able to identify healthy controls within the population as part of the samples that are collected within the biobank. So next, I'd like to talk about how we've developed different genomic platforms and genomic technologies that we've applied to samples that are collected as part of the biobank, and how we've integrated the data that's generated through these platforms into the existing infrastructure, informational infrastructure that is available to make that type of data easier for researchers to access. So the approach that the Partners HealthCare Biobank is using to be able to query different omics and genomics type data is essentially we're looking at the three main types of omics data that can be extracted. Genomics in the form of GWAS or exome SNP arrays, as well as whole genome sequencing, transcriptomics, looking at RNA-seq, looking at the mRNA expression profiles for different patients, as well as looking at the small RNA-seq or the MicroRNA profiles of different patient cohorts. In addition to that, another component of the genomic strategy is to look at the epigenomic profile of different patients, essentially through looking at methylation profiles of different patients. And so obviously this is a large initiative and will require some time. What I'd like to focus on is some of the different components that we've actually either began to integrate or we're planning to integrate in the near future. And to support these omics activities and the integration of these type of platforms, the biobank relies on the translational genomics core, which is a research core that's part of the Partners HealthCare Personalized Medicine Center. The TGC essentially is accessible to all researchers at Partners HealthCare, and it facilitates access to different omics platforms, including the ones that were mentioned in the previous slide. And the TGC, the Translational Genomics Core, has performed many different types of studies using RNA DNA for targeted and whole genome sequencing or exome sequencing, as well as epigenomic type studies either on the methyl array or by methyl-seq. And so the first component to be genomics platform component to be integrated into the Partners HealthCare Biobank is the GWAS or exome SNP array. And for this we used a multi-ethnic GWAS exome SNP array. This SNP array has about 1.6 million SNPs and is run on the Illumina iScan or HiScan system. And we're able to query 1.6 million SNPs with the option to expand that to about 2 million markers with a fairly high conversion rate for custom content. And the content of these markers query different types of different regions within the genome and the exome, and are enriched for medically relevant or clinically relevant or GWAS associated SNPs. And I've highlighted some of the markers that I thought would be interesting to cover. So these essentially are multi-ethnic exome content. It includes SNPs that are essentially found in different races and ethnicities, so that we're able to cover to understand the ethnic makeup of our cohort a little bit better. In addition, it includes a large number of GWAS markers that have a strong association with disease or have been implicated in different complex diseases. In addition, we've included markers from clinical databases or medically or markers that are medically relevant, curated from databases such as OMEM as well as ClinVar. And these essentially include functional variants in known genes that are associated with a given trait. And these include loss of function variants, variants with known pharmacogenetic relationships, as well as different human disease variants. There was a focus on essentially variants that existed within the exomes and in genes that have been associated with disease, as well as functional variants that are known to disrupt gene function or to disrupt gene expression. And so there was several different components to being able to integrate a genotyping workflow to a large initiative such as the Biobank genotyping initiative. And it's essentially, there's a wet bench component, which essentially is just how can you manage the sample tracking as well as the wet bench steps and be able to track a given sample through the different steps. And the second part was, once data is generated, how can we QC large batches that are being produced at such a high throughput and be able to flag any samples that have that are failed or any marker reactions that have failed. So to be able to track samples at the wet bench level, we used the Illumina LIMS system to essentially look at to track samples coming into the process. And we also integrated the Illumina LIMS system with our own internal LIMS system, so that we're able to track which samples were complete for downstream billing of those batches or for those samples. As for data generation, once the data is generated, essentially the files or the signals that are produced are are converted in a series of steps to genotypes. And essentially, we look at two different components of those genotypes before proceeding with further downstream analysis. And this is what's shown here in this box right here as part of the raw data QC. And what we look at is two separate components. We look at the SNP call rate, essentially for any given SNP, how well are we able to make the call at that for that marker or for that SNP. And the other QC that we look at is essentially sample-based. Are there any samples that have a large number of SNPs that are not being called properly, so we can flag those as well. Once that's complete, their downstream analysis includes different components, including essentially the generation of copy number variant calling within those within those samples, as well as imputation across the genomic data for any given sample. So to be able to capture that many number of samples and be able to QC that properly, we've developed a controlled QC report at a batch level. So that allows us to look at, instead of looking at every single sample that's coming in, we're able to kind of QC the entire batch to be able to identify any failed samples. And these batch level QC reports essentially provide a couple of things. It looks at the signal intensity versus the background intensity for the control probes across each sample, and we get an output that is shown here on the left. So panels A through G provide the different types of signal to noise ratio graphs that are used for this QC process. In addition, we look at a graph is produced to look at the SNP position call rates, and anything that's lower than a 97% call rate is essentially flagged. And the graph that allows us to do that QC is shown in panel H. In addition to the graph that are shown here on the left, a supplemental text file is also produced that actually has the list of either failed samples or failed SNPs for that sample, so that we can clearly identify the sample or SNP and follow up manually if needed on any one of these components or any one of these failed reactions. So now that we've generated all this genotyping data, the key is to be able to integrate it within the electronic medical records so that we're able to actually use this data, or researchers are able to, first of all, access this data and then use it easily and seamlessly with the existing electronic medical record data, so that we can, so that it promotes discovery or helps identify cohorts with a given genotype for different studies, as well as increases our understanding of complex diseases in terms of their penetrance and pleiotropy and other kind of genomic and complex disease type analyses. To be able to do this, the Biobank IT team has built the Biobank portal, which is shown here. And there's several different components that are communicating with each other to allow for that integration between electronic medical record data, survey data from self-reported surveys, as well as sample data and the genotyping data. And so the electronic medical records data is shown here on the upper left under the RPDR tool, which collects both the coded data as well as the text data that is provided as part of the EMR. In addition, any kind of additional research data that's been associated with a sample or with a cohort is also incorporated at this level. The third piece is the biobank data is also available as part of this larger biobank portal. And the biobank data that is included is genomic data that is produced from the arrays, but also sample-specific data. For each given patient, what are the different samples that are available, has this individual been consented for recontact, or as well as the survey data that is from the self-reported surveys. All of these are integrated into the biobank portal. The RPDR data allows for the validated phenotypes, so that a researcher can query and identify a more accurate cohort, a cohort with a larger accuracy for the disease phenotype that they're interested in, as well as the identification of controls within that within the biobank participants. And it also includes all the GWAS and the genotyping data that's been produced through the from the genomic platform. And this is all meant together. That integration is meant to drive different research initiatives by Partners HealthCare researchers. So what are the type of different query tools that are allowed by the biobank portal? Essentially, this tool integrates that those different components or different types of data into one platform where researchers can come in, define their different parameters for the cohort they're interested in, so that they can produce a list of, identify first of all, are there any participants that meet those parameters, and what is their number, and what is the type of samples that are available for those that are available in the biobank. So, for example, you can look at whether what type of samples are available. Is there DNA available for a given patient or given for individuals with a certain disease? Is plasma available or is serum available? Also, it includes the information from the self-reported surveys, the language preference, age at survey, level of education, lifestyle, as well as family history. And finally, it also includes the curated disease populations. So these are the cohorts based on either the ICD-9 codes or based on the validated phenotypes that are calculated based on the algorithm that we've implemented. And finally, there's also information on different specific types of lab results, as well as diagnoses or patient records that are that may be of interest. And in addition to that, there's also access to being able to identify samples or individuals that can act as controls for different disease states. So, for example, in this query, we're asking how many individuals, or what are the samples that are available in the biobank that have DNA that has already been collected and extracted, and those individuals have a current or past history of type 2 diabetes. And so the researchers would get an output that looks like this. Essentially, it identified 1,654 patients that meet those two criteria, and the query rate is fairly quick, only 11 seconds to be able to perform that kind of query. Another type of query is to identify disease populations that have also, for example, been genotyped. So in this case, we're looking at individuals with rheumatoid arthritis that have been genotyped on the multi-ethnic array genotyping platform, and it identified 223 individuals that we have genotyping data that have been diagnosed with rheumatoid arthritis. You can also query based on a SNP within the genotyping data. So if you're interested in a particular SNP to identify, for example, those that might be susceptible to side effects of a certain medication, or if you're looking for a particular marker so that you can identify patients that can be part of a clinical trial, the SNP can also, or the marker of interest can also drive the query. In this case, for example, we're looked at a SNP that is in EGFR and that is homozygous and is basically falls under one of these three different categories of variant types. So it's either a frameshift or a missense or a nonsense, or a loss of function start loss or a stop loss. So different types of essentially coding level type of mutations or variants. And the cohorts that we're interested in is essentially individuals that have had different types of malignancies, including lung cancer, malignant neoplasms of bronchus and lung, and different kind of diagnoses based on either the validated phenotypes or the ICD-9 codes. And based on that SNP-driven query or variant or genotype-driven query, we identified 12 individuals within the biobank population that fit those two criteria. And this is just showing that you can actually combine different diagnoses or different data from from medical records as well as survey information into one query as one parameter in the query search. So I've shown how we've integrated genomics data from SNP arrays into the biobank, and this has really facilitated the identification of cohorts and has made it easier for research to access this data. As part of our initiative, we wanted to also add another level of omics type data to the biobank portal. And in line kind of with the larger integrative strategy, ongoing efforts are focused on whole genome sequencing as well as transcriptomics type data. So we're looking at RNA-seq data as well as being able to integrate RNA-seq type data as well as small RNA-seq data. Currently, however, we've begun to investigate the use of serum and plasma samples from individuals within the biobank to be able to look at exosomal MicroRNAs analysis. So we're looking. So just a quick review of what microRNAs or small RNA-seq analysis entails. Essentially, we're looking at the role of the mRNAs in disease. mRNAs or microRNAs regulate the expression of genes, and they do that by one of two mechanisms, either translational repression or through mRNA degradation. And these mRNAs are important for a wide range of cellular processes, ranging from proliferation to apoptosis to generalized or specific type cellular functions. The mRNAs are a range of from 18 to 22 nucleotides in length, and currently there's about 2,000 to 2,500 known human microRNAs. And these microRNAs may have one specific target or several different targets but it's estimated that microRNAs target anywhere from 60 to 80% of mRNAs, and ongoing research shows that microRNAs can be useful biomarkers in disease. So one example, just to show the role of microRNAs in the regulation, in cellular regulation, as was described in the study, which essentially looked at airway smooth muscle tissue that is associated with asthma and COPD. It was identified that airway smooth muscle tissue in asthmatic individuals or in individuals with COPD exhibited hypertrophy, which is an increase in the cell size, or hyperplasia, which is an increase in the cell number through proliferation. And the several pathways are stimulated during this disease process. And essentially, these pathways are involved, or they essentially enhance cellular proliferation. And these include cyclins, CDKs, AKT, and PIK3CA pathways or molecules. And what this study did was it identified a microRNA, MIR10A, was a negative regulator of cellular proliferation in airway smooth muscle tissue through the inhibition of PIK3CA, as well as downstream inhibition of cyclins and CDK. So the idea that a microRNA can regulate a disease, can regulate the enhancement or the development of a disease state, proves that these could be a target for either as biomarkers or prognostic markers, or they could also be useful in therapeutic approaches. One class of microRNAs are circulating mRNAs, and circulating mRNAs are essentially microRNAs that have been that are found outside of the cell, and they leave the cell in one of three mechanisms that are shown here, such as through exosomes or microvesicles, or through lipid carrier molecules. And these are relatively newer in terms of trying to identify and what their function is in different cellular processes. Some research shows that they're involved in cell-to-cell communication, or they may be involved in tissue-specific regulation. Other studies show that they may be involved in promoting or inhibiting disease pathogenesis. And so circulating mRNAs can be identified in both serum and plasma samples. And the fact that you can essentially identify these biomarkers from a minimally invasive sampling, such as a blood draw, makes them advantageous to be able to be used in different diagnostic biomarker studies or prognostic biomarker studies, as well as be able to use them for predictive biomarker studies. So the fact that for our biobank cohorts we have these type of sample types. We have serum as well as plasma sample types for all patients that have consented, and we've collected blood samples from in the biobank. So one initiative was to see if we can also include circulating mRNA biomarkers type data into our biobank portal. And in that vein, we were trying to decide what is the best way to kind of approach being able to test for these biomarkers or for circulating mRNAs robustly in a high throughput fashion. And so some of the different platform selection criteria are outlined here. We would like it to be high throughput and automatable, because we're doing a large number of samples and a large number of batches, and so we want seamless integration from sample collection into the platform to be able to detect these microRNAs. We want this platform to be able to interrogate as many mRNAs as possible and to allow for novel microRNA discovery. In addition to that, given that we're looking at circulating mRNAs, and the amount of circulating mRNAs that are in serum plasma samples is very low, we want a robust wet bench workflow that will work well with low input samples. And in addition to that, we would like a streamlined post-run QC and data analysis pipeline that is able to flag samples that have failed, flag assays that have specific reactions that have failed, as well as be able to produce a type of data that can be easily integrated into the biobank platform, into the biobank portal, and the biobank larger IT and bioinformatic biobank infrastructure. So the first type of platform to be able to look at circulating mRNAs that we approached was performing small RNA-seq analysis on circulating mRNA. Here is a figure that just shows the small RNA-seq library prep workflow. It's pretty much, anyone who's familiar with this, it's pretty much a standard library prep and QC and sequencing workflow. However, there's a couple of different things within this workflow that is unique to circulating mRNAs, or that we found challenging when trying to apply this workflow for circulating mRNAs. And those are shown by the two boxes here. The first one I mentioned earlier is the input considerations. With circulating mRNAs that are extracted from serum and plasma samples, the amount of mRNAs that are extracted is so minute that it's actually difficult to quantify these using standard quantification methods such as ribo-zero or nanodrop. And so we have one of two options. We either extract and proceed into the small RNA-seq workflow, or the second option would be to try to develop a secondary assay to be able to accurately quantify these, which adds additional costs and labor to that process. The second piece is that within that extracted sample, circulating microRNAs are actually a small fraction of the total sample that is extracted. So you're getting a small amount of extraction material from serum and plasma samples, and then that small amount is only a fraction of that represents small RNA or microRNAs that you're actually interested for this type of analysis. Here are different inputs based on different types of kits that use this small RNA-seq pipeline. Most kits require a minimum of 1 microgram of total RNA, and this is if you're doing a whole white WBC total RNA extraction. With circulating mRNA, you're even getting less than what you would get with total RNA. And the second piece, as I mentioned earlier, is that it's difficult to accurately quantify using the standard methods such as nanodrop and ribogreen. The second issue or consideration for small RNA-seq library prep is the size selection step. It's critical that we're able to accurately size select for mRNA libraries and reduce the amount of contamination, whether it be adapter dimer contamination or other RNA species contamination, so that we effectively sequence the mRNA and do not minimize the efficiency of the sequencing. However, both size selection steps that are available for this workflow are laborious and are very low throughput. And the two methods I've written here. One is gel purification. Essentially, you're cutting out the fragment that represents the small RNA on a gel, or the other method to be able to size select is ampular bead purification. Both of these are difficult to automate and require manual efforts and are limited in the number of samples that you can do at one time. So this figure shows some data based on the issues that we've encountered with size selection. So in this gel on the right, on the left, the first three samples are looking at small RNA-seqs that have been libraries that have been performed using total RNA at 100 nanograms, 50 nanograms, as well as 25 nanograms. And then the second piece, the other types of samples that we've also attempted is extractions from either serum, 3 serum samples are shown here, and 2 plasma samples. And what we're looking for is a size product, is an mRNA library size product, which is around 150 to 155 base pairs. And with the total RNA extractions, we get a clear band at the 150 base pair size marker, which is the expected library size. But with both the serum and the plasma samples, it's difficult to actually get that type of band, to get that type of library yield from those type of samples. The graphs on the right are profiles of these different, for a total RNA input as well as a plasma input, and essentially this peak right here represents the small RNA-seq library at the proper size of 155 base pairs. But you can see that there's other types of contamination. These could be RNA, different RNA species, DNA contamination, as well as adapter dimers that's at 133 base pairs. So it's really difficult to actually size select for small RNA-seq or miRNA only libraries. With the plasma samples, we get the 155 base pair peak, but as you can see, it's very small compared to other peaks, including the primer dimer peaks. And other peaks that actually it's some kind of contamination that it's not clearly understood whether it's primer dimer, DNA contamination, or other RNA species. And so when we sequence this type of sample, what ends up happening is a lot of the sequencing reads that are generated represent adapter dimers rather than mRNAs, and so it limits the amount of analysis that the usability of that data that is generated from the sequencing. So what is the ideal situation to be to be able to deal with these two different technical issues? One thing that we're looking into is to be able to maximize the input of serum or plasma sample in the extraction so that we're able to concentrate these circulating mRNAs that are extracted out of these type of sample types. And the second piece is to essentially omit the size selection when possible, so we don't have to deal with the laborious workflow associated with these, and that way we also minimize the amount of yield that is lost due to these additional steps. This is the type of QC data that is generated after a small RNA-seq run. This panel here shows the number of reads. This panel here shows the number of reads per sample. All of these are serum samples, serum or plasma samples, except this one sample right here, which is total RNA. And so you can see that we actually did generate a large number of reads per sample, or variable number of reads ranging from about 2.7 million reads all the way to 14 million reads per sample. However, the question is, of course, what is the percentage of what is the percentage of reads that actually represent miRNA as opposed to contamination? The expected miRNA size after trimming and after cleaning up the reads is in the range of 20 to 25 base pairs, and we see that across all samples. So this is a positive indicator. But we also see that there's other contaminants present in each one of these samples. When we look at the different, when we look at the reads to identify what is the fraction that is mRNA versus other types of contaminants, looking across the majority of serum samples, it's in the range of 15%. As a comparison, the human brain control, the total RNA that was a library that was generated alongside as a control, generated closer to 30% of the reads were truly mRNA. Other species that are included, other contaminants that are found, as you can see here, this is the key showing that there's reads that represent genomic sequences, other RNA, and a large number of basically reads that don't align anywhere in the genome or in the transcriptome, and our theory is that these represent adapter dimers and other types of workflow-related synthetic oligos that are contaminating the final library construction. And within the RNA category, what other types of RNAs are identified? And these are shown here. So there's other small RNA species that are also represented in the sample, including long non-coding RNAs, Pee-wee RNAs, and other types of species that are currently are not being used to are not being analyzed beyond that for biomarker analysis, but could be used in the future. So this is data that has potential use in the future, but right now we're looking at the microRNA species type data. So looking at the small RNA-seq workflow for the serum plasma microRNA expression profiling, if we were to evaluate this platform, it is high throughput and automatable, so that's a plus. It does integrate all small RNAs that are found in any given sample, and it does allow for novel microRNA discovery. The wet bench workflow for low input samples, however, is one of the challenges that we face, both because of the low input issue, as well as the fact that there's a size selection step that can be laborious. And currently we're still working out the post-run QC, what it should look like, and what are the parameters that should be used, as well as the type of output that is generated, the downstream differential expression analysis type data that should be produced from these type of studies, and how to best integrate that within the biobank portal so that research can easily access that information. Another pro, however, is that for the wet bench component, as well as for the initial QCs, the small RNA-seq workflow is a workflow that we're familiar with because of other NGS library prep methods, and the QCs that are involved with that are QCs that we understand and can evaluate fairly quickly. So there are some, so right now that was one platform that we looked at. However, we wanted to kind of address the low input issue, because if we wanted to be able to offer this or to be able to run this on all biobank participants, this issue is going to come up quite often, and we wanted to explore ways that we can avoid this, the low input and serum or plasma extraction step. So one thing that we're currently evaluating at the Translational Genomics Core is a library prep system offered by HTG Molecular. The name of this system is HSeq, and essentially it facilitates transcriptional expression profiling, which could be microRNA profiling or mRNA profiling from samples with low input or limited extraction yields, which is perfect for something like serum or plasma circulating mRNAs. And the key items that are associated with this library prep system that kind of attracted us to it is that it has an extraction-free workflow. Essentially, samples are lysed and loaded directly onto the instrument. There's no extraction step. There's no QC of extraction steps. And so this helps avoid yield and biases that might be introduced during the extraction procedures. The second piece is that it's an NGS-based library, so that allows us to use existing NGS infrastructure in our lab. In addition, the fact that you're using read coverage and read depth allows for greater sensitivity and broader dynamic range compared to with other expression platforms such as qPCR or microarray. And the main piece that really attracted this system to validate the system is essentially that it does well with low input samples. So all you need is 15 microliters of plasma or serum samples to be loaded onto the system to be able to produce libraries. Just as a comparison, for small RNA-seq, we start with about 200 microliters of plasma or serum. That is extracted to yield the circulating mRNAs, and often we're not able to quantify or QC for those circulating mRNAs. The HSeq chemistry is based on a quantitative nuclease protection assay, and essentially it's a hybridization probe-based assay. So you have different RNA targets across the transcriptome, or different microRNA targets, and probes are created, or so nuclease protection probes are synthesized for those targets, and these bind to target RNA. The second step would be to eliminate excess probes and unbound RNAs using an S1 assay. And then the third step is essentially you end up with a stoichiometric pool of nucleus target probes representing the RNA transcripts or the mRNAs that were in the different samples, and these probes are actually are sequenced and then quantified to identify differential expression of the different RNA targets. So the current probe kits that are available are a microRNA whole transcriptome, which looks at about 2,000 microRNA transcripts, or 2,000 human microRNA transcripts, and several oncology biomarker panels looking at mRNA transcripts. We're discussing with HTG ways that we can develop custom panels for different for mRNA analysis of different types of pathways as well as disease states. So the workflow for the HCG HSeq is fairly straightforward. You can go from sample to expression profiling in 3 days. Again, there's no extraction step. You can see here in the very left of this workflow the different types of samples that you can put. So either a serum or plasma sample, as well as FFPE tissue, or even cell culture can be directly lysed and used as input into the HSeq system. The HSeq system essentially helps, first of all, it targets the different RNAs that are part of your kit and produces ready-to-sequence libraries. These libraries are then QC'd just using our normal or standard NGS QC analysis, and then they're sequenced using the MiSeq. And one of the major advantages, in addition to this, streamlined workflow, is that the HCG molecular also has software to be able to quantify the different probes and provide normalized counts of across the different RNA targets or microRNA targets, and in a very short time span. So the advantages of the HCG HSeq is that it's extraction free and low input, and that's kind of what what initially drove us to look into this. There is an automated workflow for either 24 samples or 96 samples, which would be advantageous for biobank samples. And there is a streamlined data processing workflow that is integrated with and is unique to the HCG probe set, or the different types of kits. The disadvantages, of course, are that it requires additional validation. This is fairly a new technology that's out on the market right now, and validations using standard and known workflows such as qPCR or MicroRNAs needs to be performed to be able to say, yes, the counts that are produced using this workflow actually match other types of data that's being produced, looking at microRNAs. And another major limitation is that it is probe-based. So you're only targeting a subset of mRNAs that are present, or you're not looking at, it doesn't allow for novel biomarker discovery at the microRNA level or at the mRNA level. So with that, I'll summarize the kind of our experience of integrating genomics platforms into existing biobank infrastructure. The biobanks essentially are a great way to advance the initiatives of personalized medicine through providing essentially two main things. One is access to patient cohorts and reducing the cost of patient samples. And to include genomic platforms into as part of the biobank, such as genotyping, such as microRNAs, requires assessment at the wet bench level, at the bioinformatic level, as well as at the IT level, to be able to integrate and streamline these processes and to be able to facilitate ease of use by researchers as well as data access. And the third component is, looking at novel assays and platforms may offer additional ways to utilize biobank samples so that we're able to identify diagnostic, prognostic, or therapeutic markers of disease. And even so, the limitation to this is that we only extract DNA, serum, and plasma, so we want to be able to maximize the usage of the usage of the types of data that is generated from looking at these samples. And new or existing assays such as small RNA-seq, such as the HCG HSeq, will allow us to kind of discover novel biomarkers using these limited types of sample types. And with that, I'd like to acknowledge the large Partners HealthCare Biobank team. Many people within different departments are involved in this large initiative that's being performed by Partners HealthCare, from IT individuals, bioinformatics teams, as well as a large lab operations team for these different workflows. And with that, I'd like to stop here, and I can take any questions.
Thank you, Dr. Amr, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Amr will answer as many questions as time permits. So without hesitation, let's jump in. And our first question asked today, are you able to extract RNA from the same blood sample used from DNA, serum, and plasma extraction?
So that's kind of one of the limitations of having one type of blood draw in one blood tube, is that we are not able to extract RNA from that same blood draw. And as part of our larger genomics strategy is to be able to do RNA-seq, and thus now we're looking into ways to having a second blood draw specifically in a tube that is to be used for that RNA extraction. So that is kind of one of the components that led us to look at other kind of transcriptomics analysis strategies, and led us to looking at circulating mRNAs as one potential option with the type of sample type that we have.
Thank you for your answer. We have a second question here today, and it asks, what type of controls are necessary for circulating miRNA analysis experiments?
Sorry, can you repeat the question?
Let me repeat that for you again. What type of controls are necessary for circulating miRNA analysis experiments?
So for circulating mRNAs type experiments, one thing that we've added is total RNA extractions. That way we don't have to worry about the low input issue, and that acts as a positive control for our experiments. However, it's not clear if there's an internal housekeeping control marker that should be found in all circulating mRNAs that are extracted from serum and plasma, and that's been an ongoing validation for us to be able to identify a marker that we can always assess to be able to establish whether a circulating mRNA experiment was successful or not. One thing that we're looking into right now is using spike and synthetic oligos that can be kind of tracked throughout the experiment and sequenced to be as a potential control, a sample-specific control, as well as a dynamic range control for circulating mRNA experiments. But that's still kind of ongoing to be able to establish what type of controls work best.
Thank you, Dr. Amr. We have time for one more question today. And this question asks, how will differential expression data from RNA-seq and miRNA analysis be integrated into the Biobank portal?
So there's several approaches that we're looking at right now into what's the best way to kind of integrate this type of data. One type of data that's generated from these experiments are essentially counts by gene. And the other piece of more kind of informative data is differential expression across genes. However, for differential expression analysis, you need a control group and a case, a case group to be able to make that kind of comparison. So one approach that we're thinking about is essentially for each individual standardizing the kind of counts, transcriptome counts or expression counts across the different genes or different circulating mRNAs that are targeted as part of these workflows. For each patient, we have those counts, and then allow the researchers to kind of create their own cases or and control groups and facilitate that comparison at the once the cases and control groups have been identified. This is of course an ongoing effort, and it certainly will be. It's unique from genotyping data because it requires actually being able to identify the case control information before the analysis can occur. So we will continue to work on this and see what is kind of most suitable for this type of data, but at the same time taking into account that researchers accessing this data come from a variety of backgrounds and expertise in this type of analyses, and to kind of facilitate the easiest way to be able to approach this the case control questions that they're interested in.
I would like to once again thank you, Dr. Amr, for your presentation. Do you have any final comments you'd like to leave with us?
So, yes, first of all, thank you for the opportunity to come and present our experience in integrating omics data into existing biobank infrastructure. And I think the one kind of final thought would be that there are a lot of tools and platforms that are available to us, and one of the key things, as you can see from this presentation, is that having those tools, having those platforms available is only the first step in being able to use it meaningfully in a larger context of the biobank. And I think that that's going to be a kind of a new experience for any type of research initiative, and will certainly change and evolve over as we discover what is the best way to kind of enhance research through biobank type data. And with that, I appreciate the opportunity to speak to you all, and I thank you for attending this presentation.
And I'd like to thank the audience for joining us today and for their interesting questions. We would also like to thank Labroots for today's educational webcast. This webcast can be viewed on demand through July 2017. Labroots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. I'll see you next time here at Labroots. Goodbye.